Chronic inflammatory enteropathy encompasses a group of disorders where dogs experience persistent and recurring signs of gastrointestinal disease. This condition is a major cause of ongoing vomiting and diarrhea in our canine companions. While the exact causes remain unknown, it's understood that a complex interplay of immunological, dietary, genetic, and environmental factors likely contributes to its development. However, a key aspect is the role of the immune system itself. Chronic inflammatory enteropathy is largely considered an immune-mediated disease, where an overactive and abnormal immune response to various antigens within the gut leads to the troublesome clinical signs we observe. Within the complex machinery of the immune system, T cells, a type of white blood cell, and the substances they produce are thought to be particularly influential in the development of intestinal inflammation. In humans with inflammatory bowel disease, a condition with similarities to chronic inflammatory enteropathy in dogs, T cells located in the lining of the intestine and the layer beneath it, known as the lamina propria, appear to have a significant impact on the immune response. Interestingly, inflammatory bowel disease in humans is also recognized as a systemic illness, affecting other parts of the body in many patients. Some researchers believe this occurs due to the migration of immune cells from the gut to other locations. While extraintestinal manifestations are not as commonly reported in dogs with chronic inflammatory enteropathy, changes in the immune cell populations in their peripheral blood have been observed. Diagnosing chronic inflammatory enteropathy in dogs involves a thorough process of ruling out other potential causes of intestinal inflammation. This is often followed by examining tissue samples under a microscope to look for evidence of inflammatory cells within the intestinal lining. Because the immune system plays such a central role, treatment often involves medications that suppress or modify the immune response. This is why the condition is sometimes referred to as steroid-responsive enteropathy or immunosuppressant-responsive enteropathy. This study highlights significant alterations in both the peripheral and intestinal T lymphocyte populations in dogs suffering from chronic inflammatory enteropathy. These findings underscore the important role of the adaptive immune response, driven by specific subsets of T lymphocytes, in the development and progression of this disease. The study also demonstrates the utility of flow cytometry in characterizing these immune cell populations in different compartments of the body, offering advantages over other techniques in veterinary medicine.